Okay, so this is the second video for the first PowerPoint, phylogenetics. So when we talk about like when and where we see deviations, and I believe some of you asked me about a multiple choice question that had an image very similar to this where the lines were not straight. What's important to remember is that there's going to be a whole different, there's going to be a lot of ways to represent the same thing. And so cladograms will not always have the same structure every time. When we talk about homologous features, we're talking about features that are very similar based on having a common ancestor. We use anagenesis, or we look at anagenesis and cladogenesis, and we determine which one happened by using those homologous features. Any feature shared by two or more species and inherited from a common ancestor is said to be homologous. Homologous features are heritable traits, which means they have to come from their DNA. What we see is that anagenesis, the traits are just going to continue on in the single species that developed from the original species, whereas sometimes when we see a division or a cladogenesis of species, two new species forming, or one species breaking away, we might see that those homologous traits are slightly different. We can also talk about the idea of ancestral versus derived traits. During the course of evolution, traits obviously change. The original shared trait is termed the ancestral trait, and the trait found in the new organisms is called the derived trait, so traits that I've inherited from my common ancestor. Any feature shared by two or more species is said to be homologous, and so we can basically say that a lot of derived traits are going to be homologous to each other. When we look at like the limbs of mammals, we all have very specific bone structures in our arms, wings, hands. And although they are very different sizes, the structure is still the same. These are all derived traits from the ancestral arm bone traits, and they're all homologous to each other. Analogous structures are going to be a little bit different. These are traits that are similar or structures that are similar but are not inherited from a common ancestor. When we look at things with wings, butterflies, bats, birds, anything with a wing does not necessarily mean that it has a common ancestor. Its function is the same, to help it fly or to help it move, but they did not come from the same species or the same common ancestor. Therefore, it's an analogous trait. And so this cannot be used to establish phylogenies, which makes sense that that a bat and a butterfly should not be on the same clade as a bird. They are going to be related to different organisms. Why do these analogous structures exist? Analogous structures evolve because of selection pressures, basically environmental pressures. It just has to do with the need for evolving to do a certain job. When we look at the right image, these are different types of burrowing moles. They both live in very similar environmental areas, but they are not genetically very closely related, and therefore they are analogous traits that they share because they do look very similar. Another reason analogous traits exist is due to evolutionary reversal. Fish gave rise to what we call tetrapods, and then cetaceans are tetrapods that return to the ocean. And so we also see that when organisms adapt to the environment, they may have originally had a common ancestor or something, but then they've moved too far away. This example, a selection pressure for flippers or fin-like structures was exerted for survival in aquatic environments. Thus, the flipper of a whale or a dolphin is very similar to that of a fish, but they are analogous structures. They don't have a lot of common ancestors, and they are only similar in purpose. Some more analogous structures. Homologous structures are coded by genes with a common origin. These genes may be mutated, but they still retain some common ancestral DNA sequence. And if we think back to our DNA coding labs, we know that the genome of the human and other animals is actually pretty similar across the board. Even when we get down to yeast's relationship to us, they are still very similar. We share about half of our DNA with yeast. Genomic sequencing essentially allows us to identify molecular homologies, or the different areas in DNA that are related. The more closely they are related, the more their DNA sequences will be alike. And I don't think I need to review this. This is stuff that we already know. The idea of the molecular clock hypothesis basically states that among closely related species, a given gene usually evolves at a reasonably constant rate. Therefore, basically, we see that evolution changes at a pretty measurable constant rate. Um, for example, the amino acid replacement for hemoglobin has occurred at a relatively constant rate for over 500 million years. If we look at the slope of the line and the amino acid differences between species as they've evolved, we see that there is a direct correlation. We see that as time passes, we see that amino acid differences in the hemoglobin in different species also changes. They are still hemoglobin proteins, but they look slightly different at the genetic level. Again, this is just a visual representation of the idea that we start with one sequence. This is the ancestral DNA. It will change just a point mutation, a letter or two, so we're still making hemoglobin or whatever protein we're talking about. As years pass, we see another point mutation or single change, so it changes just a little bit, just enough so that we're still making hemoglobin and it's still usable, but it's in a different species. The theory of molecular clocks can be used to study genomes that change rather quickly, such as the HIV virus, which is a retrovirus. Using the molecular clock, it's been estimated that HIV entered the human population in 1960, and the origin of the virus dates back to 1930. What we see is that if we can track the age of a species or an organism, we can also track at what rate they have been evolving as far as genetics are concerned. Putting this all together, so one of the most useful concepts for the application of molecular clocks is basically mutations are occurring at a rate that is regular enough to serve as a measure of how long two species diverge from a common ancestor. And so molecular clocks allows me to determine how long ago we had a common ancestor, or how long we've been evolving away from that common ancestor for, because we can assume that the less similar my DNA is, the longer I have been evolving away from a common point. When we want to reconstruct phylogenies, the following rules apply to reconstruct a phylogenetic tree. We want to look at maximum likelihood, which basically states that when considering phylogenetic hypotheses, we should take into account that one that reflects the most likely sequence of evolutionary events, given certain rules about how DNA changes over time. And so we need to look at how DNA is changing over time and what environmental and evolutionary events allow for this to occur. The other is maximum parsimony, which states that when considering multiple explanations for an observation, one should investigate the simplest explanation that is consistent with the facts. And so we should always first assume that the simplest phylogeny or relationships is the most accurate. Also, based on the differences between gene sequences in a human and a mushroom and a tulip, right, we've created two different cladograms. Tree 1 is more likely for a variety of reasons, right, based on the data, and the sum of the percentages from a point of divergence in a tree equals the percent differences as listed in the table, and so tree 1 is more likely. And this allows us to calculate that. We can talk about this in class if we would like. Making a cladogram based on traits. We have made this. You've seen a chart just like this with the ones we know that our lancelet is going to be the first species to deviate, whereas a leopard will be the last, right? Looks like this.